Mhm.
It's recording right now.
Yeah.
Good afternoon, everyone. Good afternoon. Good afternoon. I know that you guys were expecting Dr. Copeland on today, but he had an emergency. So we switched. It may be a possibility that he comes right after this lecture, and uh, and if not, though, we're gonna move right into my autocoids, and that way we'll switch. That means that if he doesn't come after the first lecture that I'm giving today, then he'll present his lecture tomorrow. All right. So sorry about that, but he had an emergency. But Happy New Year to everyone, and I hope that last year was a successful year for you guys, and you got through your exams. And so today we're going to talk about immunopharmacology and then autocoids. And we're going to make a distinction between the two, and hopefully you will see the distinction. And so, in talking about, is it? I think it's recorded. Yeah, I just want to make sure it's recording. In talking about the immunopharmacology, I want to be able to provide a basis, a baseline understanding of immunology, and so it's going to be really elementary in regards to sort of the two arms of immune function. And I think one of the most important aspects of us um, understanding immune pharmacology is the immunotherapy part of it, obviously, and then sort of the different uh, uh, groups of drugs that we use for immunotherapy. And so today's objective is actually to describe just briefly sort of the immune biology involved in, say, organ transplants, because that's where you're going to see a great deal of immunotherapy used, right, as a way to um, sort of suppress the immune system so that the individual that is receiving, say, either the transplant or the graft, it can be successful. So that's where you really use a great deal of drugs to help assist with those efforts. And then describing the functions of the different immune factors and mediators, I think is also going to be important for you guys in understanding where those mediators are positioned within the two systems that we will discuss today, and describe the different mechanisms of action of the major classes of immunosuppressants, which will be very pertinent to your exam. And then I may have some implications in dentistry towards the end. And then we'll get into the autocoids, and we'll talk about them after we talk about the immunosuppressants. Okay. So that was did it? It didn't move. Okay. Oh, so that was the objectives that I'll discuss. Uh, so the question becomes, what is immunity? Right? What are the two arms that we uh, deal with when we're talking about immunity? And basic immunity deals with the defense of the body. So it offers a level of protection against foreign pathogens, and we call those pathogens antigens. And so there are two functional arms in which we deal with, and that is innate and acquired, right? And so if we're dealing with innate immunity, we're dealing with your natural immunity. We're dealing with what is inherited. And so that requires no external stimuli in terms of the initiated response, because it's an inherited system. And then within that system, you're going to have different factors that play a role within that system that helps to assist in maybe clearance, surveillance, and defense, basically. And then you have acquired. Now, acquired is just as I said, or we call it, so there are two ways we call it acquired, but we also call it cell mediated, right? And we call it cell mediated. Anyone know why we call it sort of cell mediated? Yes, right. And you'll say so the other activating other cells. Uh, what? So this is an interesting question, and you should know it because we said we talked about it briefly, and I think in antivirals. Uh, what virus today? Uh, um, and what virus today, and what treatment that we use today deals with like cell mediated immunity? COVID. COVID. COVID is one of an example of a cell mediated response. Okay. You know, in terms of the treatment that we use, right? So because you're trying to produce what? You're trying to produce what? Antibodies against what? If it was COVID, what antibody you were trying to produce against what though? If on COVID, the spike protein, right? And so that's a prime example of a cell mediated response. Okay. So when we're talking about an innate immunity, it refers to it's a non-specific defense mechanism that come into play, and it comes into play. So when you're talking about innate immunity, is it an immediate response or a delayed response? It's an immediate response. Innate immunity is immediate response. So that tells me that you have two systems, and within these two systems, are these two systems the same or different? They're different. I mean, they're different in regards to certain elements. They're different in regards to the type of effectors, and you can see it up here, right? So if you look up here, you see a difference in terms of the effector molecules or the molecules that would be represented in terms of producing a type of response. And so when we're talking about a difference between innate immunity and cell mediated or acquired immunity, they differ in the type of effectors, they differ in specificity of antigens, they differ in speed of action, because we said that innate immunity is immediate in nature. And then induction of memory, right? So one has memory and one doesn't have memory. And so those are the type of differences that you see that overlays the system between innate and then cell mediated. Okay. So this is a backdrop for you so that when we get into say transplant therapy, or when we get into say um, asthma related events where you use glucocorticoids in that regards, you'll understand where we're coming from. Let me put this up. All right. So as I said, you're going to have differences, right? And so understanding the differences between the two, I think, is really critical. And so innate immunity really provides it's the first line of defense against any foreign substance. And that is generated through a physical barrier, physical, chemical, and microbiological barriers, as well as any type of cellular mechanism and cytokine production. And so when we're talking about cytokines, we're talking about interleukins, okay? So if we're talking about physical barriers, we're talking about skin, we're talking about the GI tract and respiratory tract, cilia, pH, mucus, floor, on the skin and in the GI tract. And so the interesting part is that even though you have this innate immunity and you have sort of these differences in effector molecules and time of response um, and and other factors, there is a complement between innate immunity and cell-mediated immunity. And what I mean is there's some overlap, and hopefully you will see it. And so in the cell-mediated immunity, you see leukocytes, macrophages, because you want to be able to, once you say kill a cell, right, you want to be able to really grab hold of that foreign substance and being able to clear it out. Macrophages allow for that process to take place. And then you have the natural killer cells, which are really prominent in helping to kill off any foreign substance as well. Okay. Okay. Let me see. Did I say okay? Did it adaptive? Okay. Okay. And here we have adaptive immunity. Uh, um, and we say it's also known as acquired. This specific immune system, it differs from innate immunity in that it is not present prior to exposure, right, to any type of antigen. And so the cells responsible for the adaptive approach is T and B cells. Those are T and B lymphocytes, okay? The T and B lymphocytes, they are developed in the bone marrow. Now the T lymphocytes undergo maturation in the thymus and the B lymphocytes undergo maturation in the bone marrow. The B lymphocytes are the ones that actually have the production of the antibodies and the immunoglobulins, right? The T lymphocytes are the one that generates secreted cytokines and also has the effect against any identifiable, say, virus or infected cell, right? So when you look at T lymphocytes or T cells, like a CD4, that's a T cell. CD8 is a T cell. Now, each one of those T cells have a different function. CD4s are the helper T cells. The CD8s are the T cells in which I said identify and lyse host cells that has been infected by a virus or other invading organism. So you can see the difference. And so the purpose is for you to understand that when we get into, say, transplantation, or when we get into, say, understanding antigen presenting cells, the CD4s and the CD8s are important in that aspect. Okay. This is just a picture highlighting the immunoglobulins or the different antibodies that are actually produced out of the B cells, right? And each one of these immunoglobulins, and I think we had a partial example of these different immunoglobulins when we were talking about, I can't remember the lecture now. I think it was in pharmacogenetics. Did I do pharmacogenetics? In one of the lectures we talked about it in regards to Steven Johnson syndrome, and we talked about it only in relationship to the IgE, right? And that response. And that response is also critical when you're talking about allergies as well. Yeah. So that becomes, so these IgEs are really important because they actually help produce sort of the hypersensitivity response that you see in patients, right? And the ability to target sort of that response with drugs is important as well. So understanding which IG system or immunoglobulin system is important helps us to subclass, right? These are subclasses, and helps us in regards to combating bacteria, viruses, and toxins. So when we're talking about bacteria, viruses, and toxins, I want to be clear, we're talking about them as antigens, right? So because they have to be presented to the cell at some point, and that's the only way in which you'll be able to sort of defeat. So that leads us to an example of what that looks like, right? What that response look like. So if you take a bacterium, right? You take this bacterium, you take antibodies that can be generated against that bacteria. Then as that antibody is actually attached to that bacteria, you have a complement of proteins that are actually produced that helps with the lysis of this bacteria. What the complement of proteins do, they actually help to like build sort of this gradient, this concentration gradient that's so high that allows for dysregulation in and out of the cell, and that's where you get the rupture of the bacteria itself. Okay, and so this is an example of how the system really attacks an antigen, or what we call a bacteria in this regards. And so you can see the same approach if we're talking about a virus as well, any type of toxin, any type of foreign substance, right? Even you see it when you're talking about, say, uh, um, if you had a transplant, right? And if that transplant didn't take, you had antibodies against that particular organ system, right? And you had it attacking, just like your body is attacking that organ system itself. Like, and so you see that in autoimmune diseases as well. Okay. So this slide highlights what I was trying to get at in regards to a sort of complement between the two. It's a general description of the immune system. It allows us to see the different type of effector. We call these effector molecules. Uh, um, we call these effector molecules. And so you see in innate immunity, you see mast cells, dendritic cells, macrophages, granulocytes are really prominent actually in innate immunity. And so you can see that as a rapid response versus adaptive immunity, which is a slow response. And so you see the difference in between the response of these two systems as well. The other difference is within the adaptive immunity, what you see is B cells and T cells. And you see the B cells producing antibodies, and you see the T cells producing its own T cells as CD4s or CD8s, and you have some other ones as well. But what you see that sort of complements that as I was talking about earlier, is the natural killer cells, where it plays a major role between innate immunity and also adaptive immunity as well, and also this sort of gamma 6 T cell as well. Okay, so you can see differences between innate immunity and adaptive immunity. I think for the sake of discussion, for us, I think a lot of our discussion is going to center around maybe adaptive immunity in a sense. Okay. So that brings us to a discussion around major histocompatibility complex. Very. So what is another name for MHC? HLA. HLA, human leukocyte, right? HLAs. So this is where, so we remember when we were talking about Stevens-Johnson syndrome? That's a drug-induced syndrome due to what? A polymorphism in a what? HLA, okay? Or a Abacavir-induced drugs, it also, Abacavir-induced Stevens-Johnson syndrome, that's also due to a polymorphism in what? An HLA. So major histocompatibility complex is like, it's an HLA. That's why you see here, it's a small region of the genome. It's highly conserved. It encodes 3 classes of polymorphic molecules known in the immune recognition unit. We call them HLAs. And then the MHC is actually located on the short arm of chromosome 6. And so the products of this MHC complex are membrane-bound receptors for the antigen and peptides, which when bound are displayed to the what? The T cell. If bound peptides are recognized by the T cells, what do you get? An immune response against those peptides is initiated. So that's how the system, right? So you have it presenting itself to that T cell, a recognition to the T cell, and producing it to the T cell. Now you get a recognition against that peptide that is being presented. Okay. And so what is important is that the immunosuppressive agents actually that we use today can be used against that system itself, right? To target that particular system. And they've been proven useful in minimizing occurrences or impacts of any deleterious effects of the immune system. And although these drugs are useful, they also have a high degree of adverse events associated with these drugs, where they've been prone to causing diseases and to increase risk of infection and malignancies itself. Right? So those are some of the things against some of these immune suppressant drugs, right? Because when you suppress the immune system, you're prone to what? Infection. Okay. So let's look at transplants. Let's look at transplants. Okay. Let's look at transplants. In regards to transplants, there is three critical relationships that we are sort of concerned with between transplant materials. And so these, you should know these three for your exam. You should know these three transplantation relationships. When we're talking about syngeneic transplants, we're talking about taking it from a genetically identical individual, usually the same individual, right? And these are similar to grafts between identical twins or isogenic strains of experimental animals. And we also can call this autologous. This is the same sort of defining principle in terms of transplantations. Then you have allogenic transplants. These are from one individual to another, right? Of the same species. It's of the same species. Okay. Now, we call these a xenograft or xenogeneic transplants. This is where I can take a tumor from a human and transplant it into, say, a mouse and get growth from that tumor. Okay. So that's a between the individuals of what different species, where I can take something from, when we're taking it from a pig, you can take the heart from a pig and they try to put it into a human, right? So that's what we call a xenogeneic or xenograft in general. In this regards, if you're doing a transplant, it'd be a xenogeneic. If you're taking it, say a tumor, taking it out, and then you sort of subplant it, subcutaneously plant it up under the skin of a mouse, we call those xenografts. Okay. So these relationships are really critical. And why are they critical? They require us to understand the MHC. All right. They require us to understand the MHC. So in addition to understanding the MHC, you have to understand what are critical problems when we're talking about transplanting. Genetic variation, because I told you uh, prior to this slide that there was what is the variation that you see in MHCs? They're polymorphic, right? A large degree of African Americans, their HLAs are polymorphic in general, right? And so you're going to have what we call mismatch. Mismatch creates a different response. It creates a deleterious response when you're transplanting between patients. So that's why you say to be a match, right? So blood type is important. And what is up there? MHC. See, blood type and MHC are the critical elements when you're talking about genetic variation, when you're doing any type of transplant. Immunological recognition of the variation is also central to actually having a successful transplant. Here, what you see two identical, these two are identical, they're identical match, right, in general. And so basically what this slide is saying that if after days of transplanting, right, after days of transplanting, as I increase in terms of the mismatch, I increase the probability of the incidence of severe graft versus host disease as I increase as your days of post transplant, right? And as you increase in mismatch, you also increase in terms of the probability of the incidence of the severe graft versus host disease. So basically what it's saying is that you would, you want a high degree of identical match between you and the transplant patient. And understanding that you will have some mismatch, right? So because highly polymorphic MHC loci in humans, they have at least one or two mismatches between you and the other patient, okay? All right. So let's move into drugs, I guess. All right. Did I am missing a slide, I think. I'm missing a slide. Am I? I am missing a slide. So the slide that I'm missing, it's really talking about, and it's critical actually. It's the immunosuppression and organ transplantation grafts. I'm missing that slide. I knew it. You have it. Okay. Okay. So in that slide, I got it. It's cool. Thank you. In that slide, you should see the original response to proliferation, right? You should see a signal 1 and a signal 2. The signal 1 is peptide response. I think signal 2 is a co-stimulatory response, if I'm correct, right? So the second response, I think, is signal 2 only, where there is removal of what? What are you removing? The what? The peptide. You see that? The peptide is removed. If you remove, so that means that you want to be able to suppress. So what are they trying to do in terms of transplanting? A graft rejection can be blocked by blocking signal 1 or signal 2. You want to block that peptide, all right, the antigen response. So that's what you see in the signal 2. In signal 1, you see blockage of what? The co-stimulatory, right? That actually helps with the signal between the antigen presenting cell and the T cell. So so you should understand that signal 2 only is a blockade in the what? Peptide. So you're using drugs to block that signal. In the signal 1 only, you're using drugs to block the co-stimulatory molecule. You see that? So that's how you reduce rejection, graft rejection, by blocking these signals. And I think we'll get into, so glucocorticoids is one of those agents that we can use to do it, and some other drugs as well. All right. So you should see immunosuppressive agents, right? Is that the one, or am I on track or no? All right. So as I was saying, these are some of your immunosuppressive agents that we're going to try to tackle today that will help us understand how we go about treatment, right? Treating sort of immunosuppressive disease, immune disease, or how we go about treating those that we want to be transplanted, right? So like glucocorticoids, cell cycle inhibitors, which you guys heard of, right? When we're talking about chemotherapeutic agents, T-cell inhibitors, drugs acting in immunophilins like cyclosporin, tacrolimus, sirolimus, cytotoxic agents. So we talked about alkylating agents. We talked about anti-metabolites, right? And chemotherapeutics, immunosuppressant antibodies, polyclonal antibodies. We use monoclonal antibodies, and then some other drugs that are also used in regards to, we talked about interferons as well in what? Antivirals. We didn't talk about opioids, but I think you guys had opioids with Dr. Sober, correct? Copeland talked opioids? Okay. Mycophenolate, and then some small biological agents. And so we won't get to everything, but we will get to enough so that you can have an understanding of the type of drugs that we use to block, say, for instance, when we talk about transplants or to treat immune diseases. immunosuppressive agents, right? When we're talking about glucocorticoids, right, we're talking about uh, um, a particular type of hormone regulated uh, drug agent, and it's given for systemic immunosuppressive effects. It downregulates the activity of antigen presenting cells and what? T cells. So prednisone or is one of the drugs that we use to it's a glucocorticoid itself. Okay, and then you have these cell cycle inhibitors, azathioprine and mycophenolate. Then you have T cell inhibitors, and these. So when we're talking about cell cycle inhibitors, we're talking about uh, um, inhibitors that do what? What if we're talking about cell cycle? What do we talk about when we were in chemotherapy? What would say if we okay? Let's take non-specific agents out of it. In this case, in this particular case, just in general, if we were talking about cell cycle inhibitors, we're talking about a direct response to the what? You say what? It's just really simple. So we're talking about a direct response to the different phases of the cell cycle. Okay, a direct response to the different phases of the cell cycle. All right. Does it make a difference where you target it in the phase of the cell cycle? Yes, it does. Right. So if you can target it in between the G0 and the G1 phase of the cell cycle, that does what for you? Because in say in the G1, we're talking about what? When you're talking about production of protein and RNA synthesis, right? You can target it. If you're talking about G0, you're talking about cells that are they're what now? They're rested. We call these cells that are in senescence. They're dormant in general, right? All right. So in this particular regard, so if we're talking about cells in the M phase, I mean, so what if you're talking about targeting cells in what? Mitosis. Okay. So it does make a difference how you develop these drugs and then what cycle. Now, ultimately, what's the ultimate function of targeting a cell cycle? It's up there. You want to do what? You want to lead to cell death. That's the ultimate fun. That's why you target it, right? But in doing so, what does that do? If you target these cells and it leads to cell death, what do we talk about in chemotherapy that happens? Say what now? Yeah, affects other, right. We talked about normal cells. We talked about the ability to cause what? Adverse reactions, because you're targeting not only the affected cell, you're targeting the what? The non-affected cell as well. Okay. So always sort of keep that in the back of your mind, because the reason why I bring that up is is sometimes doctors have a tendency to miss those contraindications sometimes, right? So although you have it labeled, uh, um, and in particular, we talked about uh, the missing of the contraindications for, say, opioids, right? For codeine, say for codeine, right? So which do you guys remember the cytochrome P450? Which what's the cytochrome P450? And codeine, the 2D6, right? Right. And so you get what? Morphine, what? Intoxication, right? So that's what I'm saying. So without really testing for that polymorphism, you can have a deleterious effect. And that goes in response to the immune system as well. Okay. So let's look at cell cycle inhibitors, right? Cell cycle inhibitors, right? We cyclophosphamide, uh, that's the CY, and as you can see, it can target any number of these cell cycles. And so in this regard, they're not really, are they specific? No, 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 no. So if you can target, if you could target any one of those phases, it's not specific. You can target anyone, right? If it's one of the ones, then it'd be specific to that cell cycle, right? So let's remember that. So like mycophenolate, the MMF, methotrexate, it's the MTX, and the azathioprine is the AZA. Okay. So as you can see, they have the ability to target, like AZA has the ability to target not only the S phase, but the G1 phase of the cell cycle. So it doesn't matter. It's not specific because you can use it in any phase of the cell cycle. I can I can use it in the S phase. It can be used in the G1 phase. It's just not used in just one of the phases of the cell cycle. Okay. Uh, then we have T cell inhibitors, anti-proliferative. Okay, don't look at the drug per se just yet. So when you're looking at sort of this proliferative response, you're looking at, and I got to remember that um, I'm doing it here, right? So what you see here, without looking at acid, which is sirolimus, don't look at it just yet. So you have an antigen presenting cell, you have it presenting sort of the peptide, you have the first signal, you have the signal between the MHC and the T cell receptor, right? And it's presenting that peptide. You get that signal. After getting the signal, you see an activation of calcineurin through the calcium channel. After that, you have the activation of the calcineurin actually um, producing what we call dephosphorylation in regards to the nuclear factor transcription, NFAT, right? So that gets dephosphorylated. That's important because once it gets dephosphorylated, it actually binds to the AP1 transcription factor, right? And so it's actually binding to that, what we call the DNA binding response element. You don't have to know all that. Just in general, I'm just trying to set up the story for you. Once it does that, now you have release of the interleukins that can actually play a role in response to the immune, it's trying to produce. So that's cytokine response, right? Trying to produce a response back to the antigen itself, right? Trying to kill that antigen or whatever, or that virus, whatever you're dealing with. Okay. So that becomes really critical, right? So you have the interleukins or the cytokines, and each one of those interleukins play a central role. Depending on which one is produced, plays a central role. That's not what we're dealing with today. What I want you to understand is now we can go back and see where in the different signals where we can attack. So sirolimus has the ability to attack at the antigen presenting cell, which we were talking about, right? So I think that becomes thinking signal 1 and signal 2. And not only that, it has the ability to target the macrophage. Okay. Now you're targeting both the antigen presenting cell and the macrophage itself. In addition to that, you have um, 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 cyclosporin, CSA, binding with one of the binding proteins or chaperone protein that actually blocks the signal between what's actually happened between the antigen presenting cell and the T cell and the activation of the calcineurin. So basically what the cyclosporin does is it blocks the activation of the calcineurin. Hint, that's critical. It blocks the activation of the calcineurin. All right. Tacrolimus does the same thing as well. Tacrolimus and cyclosporin. The only difference between the two is the response to the binding protein between the two. For the tacrolimus, you have the FKB. For the cyclosporin, you actually have the CPN. Okay. So those are the differences. But knowing that in sirolimus, you have a blockade of the antigen presenting cell and the macrophage. For the cyclosporin and for tacrolimus, you have blockade of the activation of the calcineurin. Okay, that's That's what you should get out of this slide. So as I said, glucocorticoids are a class of steroids, and they're a class of steroid hormones. Yeah. Yes. Tacrolimus. Tacrolimus. It's actually TAC, tacro, RO, limus, LIMUS. Tacro, TACRO, limus, LIMUS. I think it's in your thing, right? Yeah. So tacrolimus. So okay. So you have tacrolimus right there, and you have cyclosporin, right? The only difference between those two is the binding protein. You see that, the FKB. FKB is essential to the binding of the tacrolimus. It brings it. It's a chaperone. It brings it into the cell. You see that? That blocks that signal, right? The MHC and the T cell receptor signal in terms of the activation of the calcineurin. So basically, what is it doing? It's blocking the activation of the calcineurin. Okay. Okay. So this is I place glucocorticoids outside by itself because it's really interesting because it's a corticosteroid. It binds to the glucocorticoid receptors, as you can see here in this particular example, and you have some downstream activation of these other molecules of RAS, MAP kinases, and things of that nature. What I want you to get out of the slide is that the glucocorticoids are actually responsible for production of interleukins as well, right? And the glucocorticoids have the ability to block, like I said, the it has the ability to block like the antigen presenting cells. Okay, and I think I have a slide like that. Now, the glucocorticoids are important, as you can see from this slide. They're important in cell survival. They're important in cell proliferation and differentiation. They are important in terms of kappa beta targets. They're pro-inflammatory as well. They can be anti and pro. That's the tricky part about glucocorticoids. That's why I put them by themselves, because they can be anti and pro as well. So they can be both. They can produce and they can be against as well. You can produce a inflammatory response, or you can produce against an inflammatory response, right? And so when we're talking about sort of say a pro-inflammatory response, you see binding against different transcription factors like NF-κB, Jun, and FOS. So that's a different type of response. And then if you're talking about anti, we're talking about stopping cell proliferation. We're talking about the blockade against the MHC. Okay. So we use glucocorticoids to treat a number of different diseases: an overactive immune system, allergies, asthma, autoimmune diseases, and sepsis. Once again, we use glucocorticoids to treat an overreactive immune system, allergies, asthma, autoimmune diseases, and sepsis. Okay. Oh, okay. We talked about. So I think that's why I had it out of place, because I was putting it at the end so you can see it. So once again, you see why we use the signal blocking, what I was talking about. And here is a T cell receptor, okay, blocking sort of the T cells. And tacrolimus and cyclosporin A are ways in which we do that. And like I said, once again, you're blocking what? You're blocking calcineurin, right? And so in blocking calcineurin, you block the NFAT, right? The nuclear factor associated with T cells, you block it actually from activating transcription factors that activates the interleukins, the cytokines. So this is just what you're basically blocking that T cell, okay? And as you see here, this T cell, you have T cell receptor signaling. You don't have to know that, understand that in that T cell receptor signaling, there's a place where you have calmodulin activating the calcineurin. We were talking about the calcium, that's part of it, because the calcium acts. So this is just a much clearer picture where you saw the calcium, but you didn't see the calmodulin. So you, in that particular slide that I showed you previously, you saw calcium, but that calcium was actually producing the calmodulin, which actually activates the calcineurin. Okay, so this is just a high resolution of that slide. Okay. So here is a slide where we're talking about uh, blocking the first signal, second signal, and then third signal. And in this regard, you see in that first signal, let me see, right here you see it's resting. You see the resting APC cell, the antigen presenting cell is resting, and the OK3, which is an orthoclone monoclonal antibody, has the ability to block sort of that co-stimulatory element right there, right? Or the you have the peptide MHC against the T cell receptor, basically. So that's the first signal. In the second signal, we talked about the second signal. And we talked about the second signal in regards to what? Glucocorticoid and then sirolimus. Okay. In the third signal, you get blockade through specific antibodies. You see that? So you're blocking specific antibody, meaning you're blocking the production of cytokines or interleukins. Okay. So basically, the CD25 specific monoclonal antibodies disrupt the binding of the interleukin to its receptor. In this regard, sirolimus acts on the cytokine signal transduction sort of pathway. Okay, so basically, in like sirolimus and steroids, you have the ability to block like mTOR pathway, which is important in terms of survival or proliferation, right? You see cyclosporin A or tacrolimus actually as well over here in what? In signal one and blocking what? The calcineurin pathway. Okay. So you see two possible ways. You see the OK3, and then you see this cyclosporin A or tacrolimus in the signal one. In the signal two, you see steroids, sirolimus. Go ahead. Indirectly. I wouldn't say indirectly. I think it's just a different signal. It's maybe a downstream of that signal. It's where where you have the production of the interleukins. It's just at a much later response. So I want to show you another way in which we can look at it. Right. This is just a general way of how that response is taking place, right? It's the same sort of depiction, but just in a different response. And it's showing you how that third signal come into play. You see that? So that's why I say it's not an indirect, it's just downstream of the response. You see what I'm saying? Because in that first response, as you can see, you have the antigen presenting cell, you have the T lymphocyte, and you have, say, possibly the production of the cytokine. Then that cytokine actually going into the targeted lymphocyte. You see that? So you have the T lymphocyte, and then you have the targeted lymphocyte that you're trying to target, right? So meaning so the infected cell, right? So you targeting that lymphocyte with the what? Cytokine production. Okay. You can target against that where? with the antibody. You see that? So you can have steroids once again at the antigen presenting cell, and you can have the OK3 targeting right there, sort of the co-stimulatory molecule, right, between the antigen presenting cell and the T cell. Okay. The signal between the T cell and the antigen presenting cell. Okay. Let's take a 5 minute. Oh well. Let's take take like 2 or 3 minutes. 3 minutes. 3 minutes. 3 minutes. 3 minutes. Okay. I'm gonna try to get through it. Gotta bring it over. You gotta look at you gotta. Yeah. And now I have to put resume. Yeah. Okay. So Mhm. So you see each one have a different binding protein. Okay. So what we call them chaperone proteins in a sense, right? So you got to have something to chaperone the drug into the system, into the cell. In doing so, you got to have the ability to have it when it binds, but block, right? Because that's the purpose. What are you trying to block? Trying to block that calcium calmodulin, right? Which blocks the activation of the calcium. So the outcome of what cyclosporin A is doing, tacrolimus is doing, is what? Blocking activation of the calcineurin in the T cell. That's what they call it, a T cell block. And does that inhibit cytokines? Yeah, well, you're not going to get a response because you don't have the activation of the calcineurin to actually dephosphorylate the NFAT to actually bind to that AP1. And TAC is the same thing. It's just showing. Yeah, just showing that they have two different binding proteins or chaperones. Okay, so we're gonna next, like, I'm not gonna spend too much on here because this actually is this, right? So it's just in words. It's what's happening in T cell receptor signaling, right? In the T cell receptor signaling, you see sort of the Sharp family and inactivation, and then you see activating this, and then this DAG and the IP3, right? And then you see signal of the MAP kinase pathway that allows for activation of the transcription. And then you get the transcription IL-2, cytokine. Yeah, those drugs. Yeah. So we're gonna move I haven't started yet. When we start talking about some of the drugs, we're gonna move a little bit quick in there because we've already talked about sort of the outcome, say, of sirolimus and tacrolimus and what it's supposed to do. So I did that on purpose so we can move through. But I've given you sort of the mechanism of what it does. Okay, and because that's important. Okay. So it seems like Dr. Copeland won't be here, and so that means that I can move into the autocorticoids lecture as well. Okay. Yeah. So let's try to move through and okay. Because I originally had the autocorticoids on tomorrow, but yeah, it's actually within the same. It's at the back. It's at the back. Yes, sir. It's right after this. All right, guys, can we get started? All right. Yeah, I got it. So you wouldn't have to get up. We'll resume. Oh man, when I resume, it took me. Oh, maybe yeah, I do need your help. It took me out. It when I resumed, it took me back to.
Oh, the beginning. Let's see.
Okay.
Right here. Okay. So let's pause.
So can I have that slide? All right, now it's recording, right? All right, let's get started. Now the next slide. All right. So I want to talk in general. Let's get started if we can. The T cell receptor signaling. We've I showed you sort of description of it. This is just actually giving you a description of what we talked about in words, right? Where you have the T cell receptor activation through the Src tyrosine kinase family. That in turn phosphorylates the phospholipase C, and the activation of that phospholipase C actually leads to hydrolysis of the phosphatidylinositol 4,5 biphosphate. That generates a DAG, which is a diacylglycerol, and an inositol triphosphate. That in turn of the DAG activating the DAG protein kinase C, and that phosphorylates the RAS and GTPase, and that activates the MAP kinase pathway, which we talked about the RAF, leading to recruitment of MAP kinase cascade. And then what you end up having is a release of the IP3, the inositol triphosphate, calcium from its intracellular stores in the endoplasmic reticulum, the ER. That is what binds to the calcium, that allows the calcium to bind to the calmodulin. That's what we were talking about. That process, binding to the calmodulin, in turn activates the calcineurin. And this is in a T cell. This is what we're talking about, right? And that calcium calmodulin is dependent upon protein phosphate. And so the NFAT transcriptional regulator of IL-2 gene expression is a direct target of the calcineurin. Okay, the calcineurin, the NFAT is a direct sort of regulator of getting that IL-2 to be produced. Okay, the calcineurin dephosphorylates the cytosolic component of NFAT, and which migrates to the nucleus and induces the transcription factor. So you see, NFAT is an essential player in that process as well. Okay, because the NFAT is actually the one that is important in producing that IL-2 response, as well as calcineurin being dephosphorylated. Okay, so let's look at cyclosporin. It's a peptide antibiotic. It's an antibiotic. It blocks the action of the antigen receptor induced differentiation of the T cells, right? Inhibits the cytoplasmic phosphatase and calcineurin, which are responsible for activation of the T cell. He's here, transcription factor. So you give it orally. Just give me a little bit, okay? But we got started a little bit late. Given orally or IV, slowly and completely absorbed, metabolized by the liver. And then you see the toxicities here. Calcineurin inhibitors is a cyclosporin. Could you give me a little bit? Because we got started a little bit late today. I can, 10 minutes, 10 minutes. Give me 10 minutes. It can be used alone or in combination with other immunosuppressants, example like glucocorticoids. And it can be used alone for transplant or pancreas, kidney, and liver, also in cardiac transplants, and can be used for prophylaxis. Remember, we were talking about the prevention of graft versus host disease. Cyclosporin is one of those drugs that actually helps when you're talking about allogenic stem cell transplantation, when you're trying to reduce those signals, okay? Did I go too fast? Okay. Calcineurin inhibitors. Tacrolimus is a macrolide antibiotic. It's similar to cyclosporin. Remember, the only difference was it's what's binding to the what? Its own chaperone binding protein, which is the FKB binding protein. Okay, that was the difference. It's more potent than cyclosporin. You have similar usage in cyclosporin, especially with organ and stem transplantation. It can be given orally or by IV. It's metabolized by the liver, and you see some of the severe toxicity events that are associated with calcineurin inhibitors. Proliferation signal inhibitor sirolimus, or what we call rapamycin. Why? Because it blocks the mTOR pathway. Or everolimus is another proliferation signal inhibitor, and it inhibits IL-2 and other cytokine receptors that dependent upon signal transduction via the what? mTOR pathway. And so you block the activation of T and B cells. So sirolimus and everolimus are drugs that block activation of T and B cells. And the mechanism of action is it binds to the circulating immunophilins, the FK506BT6, blocking the action of the mTOR pathway. So it basically targets rapamycin. So we know that mTOR is responsible for a number of different elements in terms of the cell growing, angiogenesis, proliferation, metabolism. Ultimately, you have an inhibition of interleukin T cell proliferation that is occurring with this particular drug. And I'm not going to bore you with the toxicities, but they are there, and just understand them, okay? Understand which toxicities for some of the classes, okay? Mycophenolate, we talked about it in regards to it being sort of, it inhibits T and B cell responses as well, including mitogen and mixed lymphocyte. It's a semi-synthetic derivative of the mycophenolic acid, and it's derived from mold. And it is the first line of drug in preventing or reducing chronic allograft vasculopathy in cardiac transplant recipients and can be used as prophylactic agent in diseases such as lupus, rheumatoid arthritis, IBD, irritable bowel syndrome, and some dermatologic diseases as well. Thalidomide is a sedative agent that is now, it's been in clinical trials and is used in many diseases. It inhibits angiogenesis, is an anti-inflammatory and modulatory, and that inhibits the TNF alpha, reduces phagocytosis by neutrophil. It alters adhesion molecule expression and enhances cell-mediated immunity through T cell activity. Currently used in multiple myeloma. And then when we're talking about, did we use, okay. Did I skip cytotoxic drug? Okay, anti-metabolites, right? What's after that? What do you have after that? Is this the one? Thalidomide, right? I did thalidomide. And then you should have an anti-metabolite? Okay. Thalidomide? Yeah. So I'm rushing through it because you like standing. Yeah. Anti-metabolites interfere with the synthesis of nucleic acid. These include folic acid analogs. And we talked about methotrexate as a response to being what? A cell cycle inhibitor, right? Purine analogs such as azathioprine and mercaptopurine are also part of these anti-metabolites. Pyrimidine analogs, protein synthesis inhibitors are all drugs that can be used as a way of targeting the what? Cell cycle, actually. Okay. And what's that here? You know what? Because since I have you guys tomorrow, I can let him come and then I'll start here tomorrow so instead of rushing through it and let him start his lecture so he can rush through his because we don't have that much left in this part of the lecture, and that way I don't have to rush through it. But we need to start on time so I can get through because I have a meeting afterwards. So, yeah. So I'm gonna stop here, and then tomorrow we're gonna start right here at anti-metabolites. Okay. Stop it. Stop sharing.